Follow up on what Jean said now. Just before to say that I'm working with the department of Jean since till 2007, and where I'm leading a small group there, and then we are working especially on the development of novel parvovirus-based therapy. And in 2017, we'll also be the head of LOVIT. LOVIT is the Laboratory of Oncolytic Virus Immunotherapeutics, which will be based in Luxembourg, but also at the DGFZ. It's a binational strategic alliance between the DGFZ and the Luxembourg Institute of Health. And today's talk will be about our strategies to improve parvovirus-based therapies. And as Jean said, there are two main components of oncolytic virotherapy. The first component is the oncolysis that the virus is using in the cancer cells. And with the oncolysis, we have also the release of tumor-associated antigen, pumps, dumps, which is also trigger the immune system to act against the tumor. This is the important aspect because the immune system becomes the best ally of the virus in killing the cancer cells. And we are working with the H1PV parvovirus, as Jean said, and this is a very interesting virus. It's not pathogenic in humans and has a natural oncolytic, oncosuppressive abilities. The oncosuppressive abilities been demonstrated in a number of animal models, which are in part mediated by the immune system. So the virus has ability to stimulate the immune system, to elicit strong anti-cancer immune responses. So you just heard that the first clinical trial in glioma patients has been recently completed, and then the virus shows safety, is well tolerated, and shows also some promising sign of anti-cancer efficacy. So, but there's all tumors have eventually relapse in the tumor patient. That means that there is still room for improvement. And this is really the mission of the lab. So we would like to now we have some promising results at the clinical stage. So we would like to further improve the clinical outcome by increasing the efficacy of parvoviruses. And this is really is our mission. So we want to develop a second generation of parvovirus-based therapies and bring this into the clinic. And so we are working a different aspect to do that. So one logic approach is to combine the virus with other anti-cancer modalities. This is combination therapies. But also we are working on the parvovirus side to try to have the second generation of parvovirus with increased oncolytic activities. And the third, we are also trying to select those patients who most likely will benefit of parvovirus treatment to increase the efficacy. And now I will go through all these three parts. And then, so as I said, we are trying to find the drugs that could be combined with the parvoviruses. So here we don't want to change the most basic rules of mathematics, but we would like really to try to find those compounds that can synergize with the virus in killing cancer cells. And then this one, about 3 years ago, we have published that valproic acid, which is an HDAC inhibitor, which is already used in clinical as an anti-epileptic drug, could synergize with a parvovirus against cervical pancreatic carcinoma. This was a project that started really with a collaboration we had with the cancer test. It was like 7 years ago. And as you see, this is the work that contribute to this work with Jun Whaley and Serena Bonifati. We are also making combination in the lab to increase our efficacy. And the results were really interesting because here we could demonstrate in 10 different cancer cell lines from cervical and pancreatic carcinomas that the sub-lethal doses of valproicases was boosting the oncolytic activity of the virus. And here you see the virus alone. It is just one example in one pancreatic carcinoma cell line. You see the virus alone in the white bar. As soon as we bring we had this sub-lethal doses of valproicase, we can boost parvovirus cytotoxicity. And it's even more striking to see this effect on animal models. It is a xenographed animal model of pancreatic carcinoma where we implanted the SPC1 cells. And you see that in all the animals, we had completely regression of the tumors. All the animals were 100% tumor-free, and there was a long-standing effect. Was really great results under condition where single treatment did not have so much benefits for the animals. So we could also, this is published, I'm not going to mention so much about these results, but we could also study the mechanism, and we saw that the valproicase was through the modulation of an NS1 protein, which was hyperacetylated in the presence of valproicases, increased viral replication, and then also enhanced oxidative stress, and this is contributed to the increased apoptosis that we observe when we in this combo. So, but we were at the moment very much interested in glioma. There was a trial conducted in glioma, and this, when we tried to use the valproicase against glioma cells, the results were not so black and white. So it was very much dependent on the cell type of the experimental conditions that we were trying. So we were still in the need to find a drug that could synergize with the parvovirus against glioma cells. And here there was a team trying to find this magic compound, and there were some things that we have was intriguing for us. The first thing was that parvovirus H1PV is able to induce apoptosis in a large variety of cell lines, but in glioma cells, instead, is not so good, is not efficient in inducing apoptosis. Instead, induces this calypsin B-mediated cell death process that John was mentioning before. And this is good from a therapeutic point of view because the virus can overcome the resistance of the glioma cells to apoptotic drug stimulators. But it's very intriguing. Why this? Why the virus is able to induce apoptosis in some cancer cell lines and not in others, not in glioma cells? So we know also that glioma cells get resistant or are resistant to apoptotic stimuli because they overexpress anti-apoptotic BCL2 proteins. Many often overexpress these proteins. And so our hypothesis was maybe these anti-apoptotic BCL2 proteins are also the negative modulators of parvovirus-induced apoptosis in these cancer cell lines. It was logic. So we went to try it. Now there are some compounds are under development. Some have already reached phase 2, phase 3 trials, or some BH3 mimetics, which are inhibitors of these anti-apoptotic BCL2 proteins. So we tested the virus in combination with these BH3 mimetics. And here, just an example of one of these compounds, which is ABT737. And so you see here, in this, in total we tested 35 different cancer cell lines, 13 from glioma patients, all give the same results. So you see sublethal doses of this compound, BH3 mimetics, could boost really in synergistic manner the oncolysis of the virus, and by rescue the ability of the virus to induce apoptosis in this cancer cell line. So it's really suggesting that anti-apoptotic BCL2 proteins are negative modulators of parvovirus-induced apoptosis in glioma cells. And this is just an example. I don't go so much in the details, of course, could show. I have this data with me. If you are interested, I can also show you. But so we also saw that this combination is safe for normal cells. It was safe for normal astrocytes, normal melanocytes, fibroblasts, and we didn't have any effect on this. So we went to validate this in animal models, of course, and the first animal models was a challenge for the virus, I would say, because of the U87 xenograph, and this is a tumor that when we implant this ontotopically on the brain of the animals, they develop very fast. So the virus alone cannot cope because with these fast developing tumors. So indeed, you see that the virus alone was unable to eradicate the tumors, as also the ABT, the drug, ABT-737, did not manage and didn't have any therapeutic effect. These animals were dying after already 3 weeks from the cell implantation into the brain. The only animals which survived were they having co-treated with the virus and ABT-737 with a combo. And this is, again, in some of the animals, is a very long-standing effect. So it stays for the experiments for 6 months. And you clearly see on the Kaplan-Meier survival analysis that we had really a great benefit of the combo, and with a 40% of the animal completely cured. So we went to test the same combination also in a pancreatic carcinoma model. And here again, the SPC1 xenograph is also, this is not an easy model for the virus. The virus alone, you see again, it gives some benefits, but this concentration, it doesn't really help. The tumor is growing very fast. And the same again at the ABT, but the combo was really efficient in this eradication of the tumors in all the animals again. So there was after the initial growth, the tumor stopped to grow, and then there is a tumor regression. And here in this case, it was 100% survival of all the animals. Here we had to sacrifice animals because the teeth were growing too much. I don't know for what reasons, but there was 100% survival. So it's really promising, this approach. So we also analyzed the tumor samples of these animals, and you see again there is an induction of apoptosis. So, and we also study the mechanism. What is interesting from this, I don't go into the details of this, but what is interesting that is the apoptosis that the combo is inducing is an immunogenic form of apoptosis, which are all the markers for the immunogenic cell death. So calreticulin surface exposure, ATP release, secretion of HMGB1, which is also very promising. And then there is oxidative stress, calcium release, YAS stress, calcium release, and then this is also a somehow marker of immunogenic cell death. And so we know how to cure rats. We know also that humans are much more difficult than rats. Tumor in humans are much more difficult to be treated. So, but we think that this protocols deserve some clinical translation. So, and in the future, our goal is now to find new drugs that could synergize with the parvovirus. We don't stop here. We want to see if the other compounds that could even work better. And so we are going to screen now an FDA-approved compound library, and we have already the system for that, which is we will monitor in a nice throughput manner the growth, the proliferation of the cancer cells in presence or not of this virus plus drugs. And we are also, as John said, very, very much interested in exploring the possibility of combine the parvovirus with a checkpoint blockade, because we know that the parvovirus is able to convert a non-immunogenic tumor microenvironment in an immunogenic one to break the immunotolerance. So I think it would make sense now to combine the parvovirus with the checkpoint blockade. This is also something we are planning now for the near future. And so also I would like maybe here, I don't know your background, but it's a kind of call for cooperation in find more, let's say, tumor models recapitulates the human disease. One possibility would be to develop a new humanized animal model for pancreatic carcinoma, for glioma models, which have reconstituted the immune system with the PBMC from the same cancer patients. This is something we are really interested in. That's really, it would be nice to cooperate with some groups. So, as I said before, we are also working on second generation of parvoviruses. And here I will show you two strategies. Strategy one is H1PV silence strategy due to the adenoparvo chimeras that we have developed in the lab. So we were able, these people, the postdoc in the lab, Anna Paula de Oliveira, and master student, Tina Martilla, technical assistant, were able to develop a new parvovirus, which is able to express short herpin RNAs while keeping its capacity to replicate. So it's still replication competent, express short herpin RNA at a very high level, and is also efficient in gene silencing. And so this is one tool that we would like to exploit for the future. And we have done now some experiments with the gene X. I cannot now say the name of the gene for IP issue. But with this, by silencing this gene, we can already improve the capacity of the parvovirus to kill these SPC1 cells. And you see this is also in a pancreatic cancer cell line, but also in prostate cancer cell line the same. And there was also kind of validated already in vivo, where we have an increase of overall survival of these animals. So, of course, now this is a platform which is open for it's open for the targeting of other genes. So the idea is now for the future is, of course, for targeting other oncogenes, or also to target some inhibitors, immune checkpoint inhibitors, like the PD-L1. So the other approach that is also for us very, very promising is the adenoparvo chimera. And this is will be also a major focus for my lab in the future. So here we wanted to overcome, as I said before, some of the limitations of the parvoviruses, which are mainly two. Which is the first is they have a limited packaging capacity. In the parvovirus genome, we cannot insert large transgenes. It's not possible. Maximum we can insert a short herpin RNA expression cassette, 200 bases. The other limitation is low titer production. If we want to, it can be produced quite well, but not at very, very high titer. And this is on the contrary, the adenovirus can be produced at very high titer under GMP conditions that have already been established. So the idea was to insert the complete genome of the parvovirus into the adenovirus backbone, And it's like a Trojan horse strategy, that the adenovirus serves as a Trojan horse to bring the parvovirus genome into the cancer cells. And from this point, the parvovirus genome gets completely excised by the adenovirus backbone. It becomes completely independent by the adenovirus and produces parvovirus particles which are fully infectious, are normal. It was engineered the parvovirus genome in a way they can get excised for the adenovirus backbone and be completely independent, producing other virus particles. And of course, this is now, we tested adenovirus chimera, and interesting was more effective than the parental viruses in killing cancer cells. For instance, here there are cancer cells which are resistant to the parvovirus or to the adenovirus. Even when we put them in combination, they cannot be killed, but they are killed by the adenovirus parvochimera. This is the wild type. And of course, now we are interested in developing the second generation of these adenoparvochimeras. And here you can imagine there are plenty of opportunities, because we can arm now the adenovirus component. We can insert some immunostimulators, some cytokines, for instance, or some apoptosis activators like TRAIL. We can use different serotypes of adenovirus. We can retarget the adenovirus. And at the same time, we can use the same strategy also to harm. We can modify the parvovirus component, for instance, using the arming immunostimulators or RNA triggers, like the short-acting RNA expression cassette. And that can be used plus or minus other drugs. So it's like a little bit toolbox we have now that of different, we can combine different components. It's like a little bit playing with the Lego. And here in the higher expression, this is the adenovirus parvochimeras. You see that we can, the adenovirus can be harmed with pro-immune or dead strand genes containing the parvovirus genome with a short-acting RNA expression cassette, and maybe also could find drugs that could synergize together to increase the efficacy. And this is the future, our future. So in the last 2 minutes, I would like also to present you some our strategy to select those patients that most likely will benefit for parvovirus treatment-based treatment. And this is also a project that started about 7-8 years ago now. It's a long project, but it's also it's a part of our way to think science, to think about translational science. So we want, for us, what is very important is to know more about the parvovirus life cycle and to translate this knowledge in a new, more effective therapies. So we are very much trying now to understand the virus life cycle for improving the efficacy of our products. And for this, we are very much interested in studying which are the cellular factors which modulates parvovirus life cycle, and the boosters of parvovirus replication in cancer cells, because we think if we can increase replication, we will have a benefit. Also the boost of cytotoxicity. And Amit and Annabelle are working very much on that in collaboration with the project started several years ago with Laurent Brino. We have done an siRNA library screening, and through this approach, we have found possible repressor or activators of parvovirus life cycle. And we have also validated this part of these regulators. And of course, this is an important now for a source of results for us, because we would like to use these results for tumor stratification. So here we have identified these genes which are important with parvovirus life cycle. We would like to compare the list of the genes now with the genetic background of the tumors of patients in order to make the tumor stratification and to identify those patients which most likely will benefit of parvovirus treatment. So, of course, this information are also important for improving our viruses. For instance, you can imagine now that if we found negative modulators of parvovirus life cycle, we could use, by using our H1PV silencer, we could try to reduce their expression, increase virus replication and virus cytotoxicity in the cancer cells. And this is also what we would like to do in the future. So, in summary, I can make some highlights. So we were able to design a new combination therapy based on H1PV, valproic acid, and BH3 mimetics, which enhance the anticancer activity of our virus. And then also we could develop the first generation of parvovirus silencer, which parvovirus expressed in short RNAs, the first generation of adenovirus parvovirus chimeras, which is open for further genetic manipulation. And we also discovered possible modulators of parvovirus life cycle that could be predictive marker for patient stratification. And with this, I would like to thank the people which contributed to the work. I already cite some of them. And here I would like to also to thank Laurent Brino, because he's participating already in the project with the sRNA library screening. And then you for your attention, of course.